Hello, my name is Simone Randall, and today I will be presenting my part 2 of my Rubisco presentation. So first I will briefly review Rubisco's structure, importance, and how to measure Rubisco activity, and then I will be talking about how Rubisco is regulated, how it's activated, and what inhibits Rubisco. Then I'll be looking at the catalytic mechanism of Rubisco, and specifically we will be looking at the carboxylation reaction. And then finally, we will be discussing how we can optimize Rubisco efficiency. So let's get started. So as I previously mentioned, Rubisco stands for ribulose 1,5-bisphosphate carboxylase slash oxygenase, and that is because it can undergo two reactions. It could either use carbon dioxide or oxygen as its starting material to fix into the substrate ribulose 1,5-bisphosphate or RUBP. And depending on what reaction Rubisco will undergo or catalyze, it will produce different products, and the reaction, the carboxylation reaction that we will be talking about today, will produce two molecules of 3-phosphoglycerate, while the oxygenation reaction will only produce one molecule of 3-PGA and one molecule of 2-PG, which is basically useless for the plant cell or autotroph cell. So Rubisco's role in the environment is to fix inorganic carbon and turn it into usable organic carbon. And despite its very important role, it has very terrible turnover rates, and this is because carbon dioxide and oxygen compete to be the starting material for Rubisco activity. And just a reminder, I will be talking about the form 1 of Rubisco, which has the L8S8 structure, which means that there's 8 large subunits and 8 small subunits. And the 8 large subunits come in 4 dimer pairs, and between those dimer pairs, there's 2 active sites. So there are 8 active sites in this enzyme. So in my previous presentation, I spoke about a radiometric assay that can be used to measure Rubisco activity, and I just also wanted to mention that there are other environmentally friendly options and cheaper options in a way to measure Rubisco activity, and this is one of them. So you could actually use a spectrophometric assay to measure Rubisco activity, and in this article, the way they did that is they coupled the production of 3-PGA to a change in absorbance, and it's associated with the oxidation of NADH. And they actually measured the Rubisco activity in 3 different reactions that couple the oxidation of NADH with the production of 3-PGA, but they use different enzymes in between. So moving forward, I also had this image on my last presentation, and I kind of briefly skimmed over the carbamyl lysine 201 residue, and that's what I'll be talking about today. So actually, it's really important in the activation of Rubisco, because typically when Rubisco is in its inactive state, Rubisco will have a inhibitor sitting in its active site instead of magnesium, and the inhibitor can actually be its own substrate, so that's pretty interesting. So ribulose 1,5-bisphosphate sits in its active site, but I would also like to mention that these are also the other naturally occurring inhibitors of Rubisco. So as you can see, there's RUBP, and then there's all these other natural inhibitors. And it's really interesting because they resemble very closely to these intermediates that can be found in the carboxylase reaction. And I will also point it out when I'm going through the catalytic mechanism, but that's just another interesting fact. So sitting in the active site is a inhibitor, and it is blocking the lysine 201 residue from being carbamylated. And what removes the RUPB or inhibitor from the active site is this enzyme here called Rubisco activase. So Rubisco activase actually couples ATP hydrolysis to the removal of the bound RUBP or inhibitor, and then it exposes the lysine 201 residue, and the lysine 201 residue can then be carbamylated with another CO2 molecule that's not going to be involved in the carboxylase reaction. And once the lysine 201 is carbamylated, it puts a negative charge on the lysine 201 residue, and this immediately makes magnesium 2 plus ion attracted to the lysine 201 residue, and that's what makes magnesium just go on and sit in the active site, and of course interact with all these other residues and oxygen molecules of the substrates. So let's take a look at the mechanism. So sitting right here is Rubisco's active site with the magnesium interacting with the the lysine 201 carbamylated residues, and it's also shown to it's also showing ribose 1,5 bisphosphate. And what actually initiates this reaction is the tautomerization of RUBP to the enodiolate intermediate. So the enolization is initiated by the deprotonization of this C3 carbon, C3 position of the RUBP molecule. And what deprotonates that carbon is speculated and debated, but due to steric constraints observed in the crystal structures of Rubisco and of this process, it's highly plausible that it's the lysine 201 residue that acts as the enzyme base that deprotonates the C3 position of RUBP. And then once that occurs, carbon dioxide will be added into the active site where it will be interacting with magnesium 2 plus that's actually sitting in the active site. And what magnesium 2 plus does is it polarizes the carbon dioxide, and it gets it ready for a nucleophilic attack by the enodiolate intermediate. And you can also see here this histidine 294 residue is also acting as a base and deprotonating this hydroxyl group off the C3 of RUBP, or the enodiolate intermediate. And what this does is it will form a beta keto acid intermediate. And this is the intermediate that I previously showed as a naturally occurring inhibitor. So it's not the inhibitor, but this is what all the naturally occurring inhibitors kind of mimic. So that's pretty interesting. And then in this reaction, a water molecule comes in and hydrolyzes the beta keto acid intermediate. And what that does is it will create this gem diol intermediate seen right here. And then finally, the gem diol intermediate is actually cleaved in this between the C2 and the C3 position right here. And this will produce one molecule of 3-PGA. Then the other molecule is actually a carbanion, and this lysine 175 residue will protonate the anion seen right here, and then it will produce the 3-PGA. So we will get, two molecules of 3-PGA, which will continue on into the process of making ATP for the autotroph. So, So I found this article on optimizing Rubisco efficiency, and obviously this is a very hot topic because Rubisco is kind of very important for our daily lives. It's also important to for the optimization of like crop productivity as well. So in this article by Perry et al., they suggest manipulating Rubisco activase and the abundance of its naturally occurring inhibitors. And the way we can manipulate that is by targeting their synthesis or degrading them. And in this way, we can modulate the Rubisco activity, and then also we can stabilize Rubisco under stress. So this leaves us with kind of like a glimmer of hope, because there's a lot of research going into this right now. And I wanted to just finish up with a summary, and of course I hope that you guys learned how important Rubisco is, a little bit about its structure, where it could be found, how it's regulated, and what catalytic mechanism it undergoes, and some assays to measure its activity, and a method to optimize Rubisco efficiency. So this is my works cited, and let me know if you guys have any questions that you would like to discuss. Thank you.